So my talk will be about storage and flexible retrieval of sequences in recurrent networks. And okay. So one of the most fundamental questions in neuroscience is to understand the mechanisms of learning and memory, and to connect this with the theme of this workshop. This is a quintessential multi-scale problem, because learning occurs thanks to modifications of synaptic connectivity. So it's phenomenon at the subcellular level that is driven by pre- and postsynaptic activity and also neuromodulators. And it is thought that memory persists thanks to the maintenance of these modifications. However, to understand learning and memory, it is not enough to understand how synapses changes their state, because learned behaviors are produced through the dynamics of very large networks whose connectivity has been shaped by synaptic plasticity. So we need to understand what's happening at the single synapse level. We need to build mathematical models that describe what's happening at this level, but we need also to understand how plasticity changes the dynamics of large networks. And so, of course, we know a lot now about synaptic plasticity. There have been decades of research characterizing the phenomenology of plasticity, mostly in vitro, but now increasingly also in vivo. And in particular, it's been shown that the activity of pre- and postsynaptic neurons can modify synaptic strength in specific ways. And a lot of attention has been devoted to how the temporal difference between pre- and postsynaptic activity can change synaptic strength. And something that's not very widely appreciated is that there is a large heterogeneity in how how this timing difference affects plasticity. So in some cases, people have found that plasticity is temporally symmetric, in the sense that if you have coincident pre- and postsynaptic activity at the scale of maybe tens of milliseconds, then the synapse tends to strengthen, but it doesn't matter whether pre- or postsynaptic neurons fire first or second. So this is the case in CA3 slices. This is work by Peter Jonas. Right. In other cases, the sign of delta t, this timing difference between pre and post, matters a lot. Like in this case, which is now in hippocampal cultures, where you see that if pre fires before post, you have strengthening of synaptic plasticity, while if post fires before pre, then the opposite happens. You have a decrease in synaptic strength. So this is a temporally asymmetric form of plasticity. There is a strong heterogeneity in the temporal symmetry of plasticity, but there is also a heterogeneity in the time scales of plasticity. In fact, while these experiments were done by pairing single spikes of pre and postsynaptic neurons, you have a different forms of plasticity that was recently discovered in the hippocampus called behavioral timescale plasticity, where now you pair a plateau potential of the postsynaptic cell with a burst of presynaptic activity. And then this leads to plasticity at a much longer time scale. So now you can see that you need a coincidence now at the scale of seconds, no longer at the scale of tens of milliseconds. But again, you can find these different forms, some more temporally symmetric, some more temporally asymmetric, during CA3 and CA1. So one first question, as I mentioned in the first slide, is basically what kind of models of synaptic plasticity can capture this data. And this is a question that have interested me for a long time. So with a graduate student, Michael Graupner, more than about 15 years ago now, we built a model that was where we try to connect, biophysical mechanisms of what's going on at the synapse with a simplified. We try to implement these mechanisms in a highly simplified model. So briefly, this is by now a classic model for what's happening at glutamatergic synapses onto pyramidal cells, both in hippocampus and cortex. And the idea is that to trigger plasticity, you need first to have a calcium influx into the postsynaptic spine. And this happens through NMDA receptors. There are also voltage-gated calcium channels that that are not shown in this slide. And then this calcium entry triggers a number of things, basically activate calmodulin and then CAMK2, and then there is a cascade of things that happened until at some point new alpha receptors can be inserted at the synapse, or these receptors can also be phosphorylated, and both mechanisms tend to strengthen the synapse. And vice versa, you can have dephosphorylation of CAMK2, so inactivation, which would lead to depression, basically removal of these receptors or dephosphorylation of these receptors. All right, so of course these are highly complex phenomena, and we'd like to find a kind of minimal model of this phenomena, and so that's essentially what we did with Nicolas Brunel. So we built this kind of minimal description of plasticity, where the single variable that controls plasticity is just the calcium concentration in the postsynaptic spine. And the idea is that calcium triggers trigger calcium transients, so both pre- and postsynaptic spikes. So this is the dynamics of calcium in response to these spikes. These are simple jumps followed by exponential decay with sometimes some tau Ca. And then what we do is we add some thresholds for plasticity, so a high threshold for potentiation and a lower threshold for depression. And the idea is that the presynaptic transients capture calcium entry due to opening of NMDA receptors, while postsynaptic transients will capture calcium transient due to voltage-gated calcium channels. And we build various variants of these models, but I don't have time to tell you about it. The main point here is that you have these thresholds where the synaptic efficacy itself will basically be driven by the time spent by. So when the calcium concentration exceeds this threshold, the efficacy goes up, while when it's in this intermediate region, it goes down. And what we did is study the behavior of this model in response to a classic spike time independent plasticity protocol, right? And so that's the protocol that people use in these experiments. First, you just stimulate pre to estimate baseline weight, then you pair pre and post with some specific delta t, and then you check at the end, you just stimulate post to just estimate the synaptic strength after the pairing. So in the model, we have basically, these are the possible behaviors of the so-called STDP curves that tell us how the strengths change as a function of delta t, this parameter, basically the difference between post and pre. And basically, you can see that in the space of parameters of the model, in particular the amplitude of the calcium transient C pre and C post, you can have different behaviors. We can have this behavior where you have depression at negative delta t, followed by potentiation at positive delta t. So this is a strongly temporally asymmetric curve, and that happens when C post is larger than C pre in that area here. But we can have also regions where the curve is temporally symmetric, and trivially it's temporally symmetric if C post and C pre are the same, right? Then it must be the curves must be symmetric, as in this case. So this looks like CA3 plasticity in CA3. This looks like more plasticity seen in the other systems like hippocampal cultures, for instance. But you have also these other types of curves that can occur, the opposite, the reverse STDP, only depression, or curve which is called the depression, potentiation, depression. So two depression windows surrounding the potentiation. And so in the model, we can understand what controls the degree of symmetry of this curve. And of course, we can understand also what controls the width of this curve, the time scale, because it's basically controlled by the time scales of these calcium transients. And for instance, in the case of BTSP, we expect that it will be much longer because these plateau potentials are very extended nonlinear events that can last for a second or so. All right. And interestingly, all these different curves have been actually observed experimentally in different systems, or sometimes even in the same preparations, but by either different groups or sometimes by the same groups by varying parameters like extracellular calcium concentration. So, and to my knowledge, I do not know of any different types of curves that have been seen experimentally. All right. So that was just a very quick description of this model. So there are, of course, much more that you can find in published papers. So we've shown that the model can reproduce quantitatively the dependence of plasticity on spike timing, firing rate, and also different types of spike patterns like triplets, quadruplets. And I want to emphasize that most of these data cannot actually be captured even qualitatively by classic pair-based STDP rules that have been used a lot by theorists. So this model, which kind of goes one step towards biophysics by implementing some landmark features that are observed in experiments, goes a long way towards capturing quantitatively all this data that cannot be captured by STDP models. Furthermore, it's simple enough to be implementable in large-scale simulations. And finally, this model allows you also to make experimental predictions. For instance, what happens to plasticity when you vary parameters like extracellular calcium concentration? And we've verified some of these predictions experimentally in hippocampal slices, collaborating with a group in Marseille, Dominique Deban. All right, so now let's jump to the network level. And so I'll be switching gears a little bit. So the question, again, is basically how does plasticity shape the dynamics of networks? And of course, there are several classical scenarios that we know about that have been around for a very long time. For example, a scenario in which plasticity shapes the network in such a way as to create fixed point attractors. So this is the Hopfield model scenario, right? So you're using, in this case, a very symmetric form of plasticity. So you need to create a connectivity matrix that is close to being symmetric, not necessarily perfectly symmetric. And so basically you create these strong excitatory feedback loops among groups of neurons. And so that leads to the creation of fixed point attractors where these neurons can be active while all the rest of the network remains at lower rates. Right. And that, of course, can be helpful to maintain information in short-term memory. So it's been a kind of classical model for working memory. All right. So that can typically be created by temporally symmetric plasticity. Now you have a radically different model where instead you use a temporally asymmetric connectivity, and then you can see that you can shape the connectivity in a fundamentally different way if you have time-varying inputs that activate in different groups of neurons in temporal succession. So suppose you have an input that drives these neurons first, and then these groups of neurons later, and so on and so forth. And so if you have a temporally asymmetric plasticity rule, of course, you will generate a very asymmetric connectivity matrix. These connections will be strengthened, but the connection in the other direction will go down because they will be depressed, right? And so that leads to something that's sometimes called functionally feed-forward connectivity, or it's an example of an asymmetric connectivity. And now what can happen is that following this this shaping of the connectivity matrix, you can have a network that can generate autonomously these sequences. So if you initialize the network in the appropriate state, like you activate these neurons, then the network will be able to recall the sequence of states thanks to the modified connectivity. And this clearly can be helpful for a network that is needed to generate temporally structured behavior. Suppose that you need to generate some temporal structure, like to control some particular movement, then this would be clearly very useful. So now in the second part of the talk, I will focus on this scenario. And this is, so the phenomenon of sequential activity is a phenomenon that's been observed in many different structures, during many different behaviors. And a very common behavior, of course, is again generation of temporally structured motor, temporally structured movements. A beautiful example that probably many of you know is the example of a system in the bird, in zebra finches in particular, that is responsible for generating songs. And when you record neurons in that structure, the area HVC, while the bird is singing, then you see that different neurons fire at different times during the song. So for instance, this neuron fires at the very beginning, and then this neuron a little bit later. And so this is basically one of the earliest papers documented this phenomenon, but now people have recorded many more neurons, and you see that basically you have neurons firing at any point during production of the song. In this case, note that activity of neurons is extremely sparse. Most of these neurons just fire a single burst, a very brief burst. Notice the scale here, about 10 milliseconds of a few spikes, and that's it, right? Not all of them. This one is a little bit, fires a little bit longer, but this is the typical behavior of most neurons. You have other, you see also sequential activity in other contexts. And a very interesting one is basically when an animal needs to keep track of time. So this is this is the example of time cells in the hippocampus. So this is from an experiment by Eva Pastalkova in Yuri Bujaki's lab, where they trained mice to run in a wheel for about 15 seconds. And after 15 seconds, the mice learned that after that could go to some water pot or to drink or some food location. I don't remember what was given to the mouse. So while the mouse is running in the wheel, what you can see is that different neurons are active at different times. So you have these neurons that fire at the very beginning. You have actually a whole group of neurons firing quite early, but then being mostly silent, though not all of them. Some neurons tend to fire kind of persistently, so you have a much more heterogeneity and diversity here. But then you have other neurons that fires at different times, and you can find neurons basically that are active at any given time in this during this interval. Note that this is far more heterogeneous than in the HVC system. You have some neurons that fire for seconds, others that are very brief, that are active on the other short time, like this one. But you can see that from the activity of these neurons, you can decode the time that has passed from the beginning, from the mouse started running, and so presumably that's the mouse uses these neurons to figure out when it can stop and then collect the report. All right, so basically you can see that, these are just two examples, but there are many others. In all these areas, you can see that we have sequences of firing cells that tile the entire time interval. That doesn't mean that cells fire at just a given time point. Some cells can fire for a big part of the temporal interval or can have multiple time fields. This really depends on the area and the behavior. You have this strong diversity of sparseness levels and temporal profiles on neuronal activity. And also there's been work where people have shown that you can generate multiple sequences in the same area. So depending on the context, you can have multiple sequences that are generated depending on for example, the decision that the animal needs to make. All right. So again, this is of course the question of sequence storage and generation has been studied by many groups, starting actually by a pioneering paper by Shunichi Amari, who was also a precursor of the Hopfield model, and then work by Kleinfeld, Sompolinsky. So this early work focused on networks of binary neurons. Then you had a wave of papers exploring the syn-fire chain hypothesis, where now you have groups of synchronously firing neurons that trigger the activity of the next group, and so on and so forth. Syn-fire chain models actually seem to be a good description of what's happening in the bird, right? In the songbird, where you have these very brief and periods of activity of each node. However, these models do not really capture the diversity of dynamics that we see in the data. In particular, none of these models could reproduce this particular, what you see in this slide. And then another strategy has been more recently to train networks using supervised learning algorithms. So they can, and of course, using a supervised learning algorithm, you can train a network to do pretty much anything, right? But the problem is that these learning algorithms are fairly implausible. So back a few years ago, actually this project started when I was still at the University of Chicago. A graduate student of mine started to reconsider kind of standard rate models, and also he used spiking networks, as I will show briefly, and just wondered what would happen if you just used kind of a very simple temporally asymmetric Hebbian plasticity rule. And we'll see that this goes a long way into capturing the diversity of dynamics in the data. And furthermore, there is the added bonus that these models are also analytically tractable. All right, so what is the model that he considered? So this is kind of a vanilla rate model, if you will. So basically the rate variables each neuron described by a fine rate Ri, decay term. This is the transfer function, and then these are the external inputs to the network, IIX, X for external, and then these are the recurrent. This is the recurrent connectivity. The transfer function, you could use pretty much anything. It turns out that this function is quite helpful to simplify calculations. It's basically the cumulative, proportional to the cumulative distribution function of a Gaussian. With parameter theta, it's a kind of threshold, and sigma is the related to the slope here. And of course, the crucial point here is how you structure the connectivity matrix, right? And so here we will take actually a model that is very, very related to models that have been used by physicists in the late 80s and 90s, where the idea is that at each point in time in the sequence, the network is driven by some external input to a particular state. So this is basically this size here are IID random variables. So they are independent for each neuron and for each time step in a sequence. For simplicity, we discretize time, though we also looked at the continuous case. But it's kind of simpler to kind of discretize the sequence. So the idea is that psi IST is describes how an input at time t for a sequence S activates neuron I. Okay, so that's it. And then, so how do you modify connectivity using this from these sequences? Well, you use basically a simple rule that where the connectivity matrix will be modified according to the product of the activity at time t+1 of the postsynaptic neuron and the activity at time t of the presynaptic neuron. So that the t+1 here will basically lead to this temporal asymmetry, right? If you remove t+1, then you get a symmetric connectivity matrix, and this is essentially the Hopfield, the learning rule in the Hopfield model. And then, for simplicity, you just sum over all time steps over all sequences. Well, these are just normalization constants. For variety of reasons, we do this on top of a random Erdős-Renyi matrix with a small connection probability, and that's helpful for the calculations actually. So basically, Cij is just a random 0-1 matrix, and connection probability is very small. All right, and again, the motivation here is that, what is this t+1? +1 here is meant to basically is the time difference that maximizes potentiation. So in this case, it would be 10 seconds, but it can be depending on the system and on the type of synapse, it could vary. All right, so there is one case where actually we can do a lot of calculations, which unsurprisingly the case when we have Gaussian patterns, right? So the size are we draw size from a standard Gaussian, and so what happens in this case? Well, and I don't have time to talk about the continuous case, but let's focus on discretized case. So this is a particular sequence, and basically this indicates the values of this random variable for one particular neuron at different times. So it's just a bunch of IID standard Gaussians. Now, after you structure the connectivity, you initialize the network using an external input that drives the network towards its the first step in the sequence. And then what happens is that the network goes through some trajectory where the rates of each neurons kind of try to catch up with the are driven by disk size. So when the size are high, the rate of these neurons tends to go up, and then it goes down here, and then it will go up again because again you have some this size were high towards the end of the sequence. So in this case, you have a neuron that has kind of a two time fields, if you will. It fires at two different times in the sequence. But then, of course, because all the neurons see random IID sequences, basically of this size, you can see that there is a strong heterogeneity. Some neurons will just have basically just are active at the beginning, others will be active in the middle, and so on and so forth. We can quantify, so there is a strong heterogeneity in the temporal patterns of activity. We can quantify how well the sequence is retrieved by measuring the overlaps of the network states at any given time with each of these patterns, right? And so when we look at these overlaps, well, the idea is that initially the overlap with the first pattern is strong because the external input is pointing towards this particular pattern. So that's the first curve here, the darker one. And then this one decays, but then it triggers the overlap with the next pattern, rises and peaks and goes down, and so on and so forth. So you have a kind of wave. So these overlaps get increase one after the other before going down. And the fact that the sequence is retrieved is that basically you, at any point in the sequence, you have the overlap with one of the patterns, the sequence that is high. And so basically just a quick a slide on mathematical methods and basically that also gives us some intuition about what happens. So there is one specific case which is analytically tractable when basically the patterns size are Gaussian. In this case, you can actually derive in the large N limit equations for these overlaps. And these equations look remarkably simple, right? So if you look at the overlap with the pattern mu, what you can see is that there is this decay term, but then it's driven by the overlap with the previous pattern in the sequence, right? So when the network is correlated with one pattern, then it will tend to drive the overlap with the next pattern in the sequence. There is this function here, g, which is a kind of gain function, which depends both on the norm of the overlap vector, the vector of all overlaps, and then here there is a kind of interference term, which is similar to what you have in the Hopfield model, where you have interference between different memories that can affect the retrieval of a particular pattern. Here it's similar, except that this is interference with different sequences that are stored in the network. This gain function has actually can be described by a simple equation, where phi here is the transfer function. Actually, all the complications are actually in the dynamics of this term here, m of t, which is given by a couple of integral differential equations. And by the way, these are standard techniques from dynamical mean field theory that allow you to derive these equations. Interestingly, when you can see that this gain function, when you retrieve a sequence, it becomes approximately constant during and hovers slightly above 1. And you can see clearly that if g is much smaller than 1, the overlaps will decay, so the sequence won't be retrieved. And of course, g cannot be much larger than 1 because otherwise these overlaps will explode, which cannot be, given that the activity of neurons is bounded. So, but basically, if g is constant, then these equations become incredibly simple, right? And you can basically solve them analytically, and you see that each overlap will be given by this equation. Basically, it's an exponential function times t to the power mu minus 1. And in particular, what it tells us is that the width of the overlaps is actually growing progressively fine. So you have a pretty short overlap with the first pattern in the sequence, but then progressively you have this growth of the width as square root, proportionally as the square root of time. And this is something that we see here, right? We see that the width here is relatively narrow, but then it becomes broader and broader. And this is actually reflected also in the time fields themselves. When you look at the at the width of the individual neurons time fields, they tend to become broader and broader. And so that's another way to look at it. You can see basically this is now all neurons in the large network. The neurons are sorted by their by the time of their peak firing rate, and you can visualize this strong heterogeneity of profiles of activity. You can see that if you plot the width of the time fields, they become broader and broader as time goes by, and the width, in fact, which is predicted to go as square root of t, matches quite well with the simulations. And interestingly, that also matches, at least qualitatively, very well some data, in particular the data about these time fields, where you see clearly that initially these time fields are pretty narrow, but then they become broader and broader with time. And this is basically the data. So in that paper, they've basically described it as a straight line. This model would predict that it's better captured by a square root function. Also, something that the model reproduces automatically is that you have a higher density of time fields in the initial phase of the sequence than towards the end. As you can see here, you can see a big fraction of neurons firing initially. And note that this is not something that we put in the model. That just comes out of the dynamics. This is not at all something that you could have guessed from looking at the model initially. Okay, we can also do things like computing storage capacity. So there is a long tradition, especially in the statistical physics community, of looking at the capacity of different models. In particular, the capacity as expected scales the total number of patterns that you can store, which would be the number of sequences times the number of time steps in the sequence, scales as the number of plastic connections per neuron. This is a kind of general result in this class of models. But interestingly, it depends strongly on the shape of the transfer function, both the threshold and sigma, which is the slope parameter. Interestingly, it peaks here when theta is relatively high. So that means that the activity of the network has to be relatively low. And also, it's not maximized for a step function, but rather there is an optimal slope of the transfer function that maximizes capacity, which is this region here. And these lines are very simple, actually, very simple equations describing these lines. It's just constant, and this is also has a very simple equation. Okay, so this was for Gaussian patterns. So now we can use also different, disk size. We can use different distributions for disk size. In particular, we can use instead sparse patterns where the psi can take a value 1 minus f with some probability f, or minus f with probability 1 minus f, right? And f with f being very, very small. The idea is that in subsystems you expect neurons to be active only with a small probability. And then, of course, that's entirely to be expected, right? But the sequences then becomes much sparser as you decrease f. So this is a case where f, I think here was 0.05. You see now that basically neurons, the width of these time fields becomes much more narrow. And so you can, when you decrease f, you would tend to go towards the limit, kind of the HBC limit, where neurons become very, very sparse, or kind of thin-fire chain-like limit, if you like. All right, and just I kind of I forgot when I started and how much time I still have.
10 more minutes. You have 10 more minutes.
Oh, perfect, fantastic. Okay. Yeah, very briefly. So everything I told you about so far, it was using this very simple rate model. But you can actually, guided by the results in the rate model, you can also build spiking network implementations that function essentially in a similar way. And so Max built kind of this in 2 steps. He started by using a rate network with separate excitatory and inhibitory populations. Note that in the excitatory inhibitory network, only the excitatory connectivity is shaped by learning. Though another student at Duke also looked at cases where inhibitory connectivity is also shaped by learning, but I don't have time to talk about this. So in fact, inhibitory neurons are just here, they're just used to stabilize network activity and to ensure that you don't have, the whole network doesn't explode. And finally, we can go to the next step and also use a spiking network, basically network of kind of standard excitatory and inhibitory leaky integrated fire neurons connected randomly, but with the excitatory to excitatory connectivity matrix that's shaped using the rule that I described earlier. And again, here you see that inhibitory neurons are basically firing. It's a kind of blanket inhibition scenario. They are just firing all the time and not in any specific way. I think there is some kind of evidence in area HDC that this is what inhibitory neurons do. All right, so now let's come back for the last 5 to 10 minutes. Let's come back to this issue of the head. And sometimes we see asymmetric plasticity. So we actually wondered what would happen if you had in the same network neurons with these different plasticity rules, right? Or it doesn't mean to be a same network. It could be two different networks that are sufficiently strongly connected together. So what's the effect of heterogeneity in synaptic plasticity rule in a network? So basically, we can understand pretty much what will happen by just looking at this simple cartoon. So I should just start with this simple cartoon. So the idea now is that you have these neurons that some neurons have asymmetric plasticity, so the A neurons, and then others have symmetric plasticity. So let's see what should happen now if, again, we consider the scenario where we have temporally varying inputs to this network. So they drive activity of different groups of neurons in temporal succession. So let's say you have a first group of neurons that get activated at some time point t, and then another group later, the blue group later, right? And this is the resulting connectivity matrix. You see that the symmetric neurons, they just want to connect. They just want to connect together, right? The red neurons, they want to connect with the other red neurons, so they are. But they also They also want to get inputs from the A neurons actually, because it's really the postsynaptic identity that determines the plasticity rule in this case, right? And then same for the blue S neurons, they want to connect together. But now the blue A neurons, they don't want to connect with the same neurons. They want to connect. They want to receive inputs from the neurons that were active previously, so from the red neurons. So you have these links that are strengthened here. So why is this interesting? So, well, now suppose that you have external inputs that you can target these two different groups of neurons in a different way. You could have different inputs to the S neurons and then to the A neurons. Well, if you shut down the A neurons in a kind of extreme case, then basically you can have perfectly stable fixed point attractors where these guys are, they just keep firing. So this is the standard persistent activity scenario. Now, vice versa, you shut down these S neurons. Now the activity will move basically from the red state to the blue state, and so on and so forth, right? So you will tend to move along the sequence. So in a way, these the A neurons are a bit like accelerators, right? And with these inputs, you can, it's like pushing on the accelerator pedal, and the S neurons are more like the brakes. They want to stay where they are. And in fact, this exactly works as I described. You can very robustly, in particular, change the speed of sequence retrieval by varying, the different inputs that you're providing to these two populations. When you have IA is the input to the A population is stronger than IS, then you will go through the sequence pretty quickly, as you can see here. But vice versa, you can slow down the retrieval by increasing the inputs to the S population, as in this case. So this is exactly the same network here. I've just changed the strength of the external inputs to these two populations. And you can also, switch, as I already mentioned, switch from a fixed point attractor to a sequential activity. So this is a case where you initialize the network in the first state of the sequence, and then you have a strong input to the population 2 here are the symmetric neurons, and so it's perfectly stable there. But then at some point, we change the inputs, and then you're going through the sequence. So this could be like a simplified model, if you will, from a preparatory state and then you're kind of preparing for execution of a particular movement, and then you change the inputs, and then maybe you have input from the thalamus that tell you now it's time to go. Note that this is not a specific input to that particular sequence; it's just a generic input towards the asymmetric neurons. There is nothing specific towards this sequence, but because you're in this particular state, then you will trigger the appropriate sequence. And you can do that at different speeds by modulating the strength of this input. And in fact, what you can see here is that when you look at the activity of these neurons when you're executing this, when the sequence is retrieved at different speeds, you have a kind of approximate temporal rescaling of the time fields of these neurons. They tend to be broader, but it's not exactly temporal rescaling. You see that this neuron kind of had a higher firing rate for the short interval, but now it's smaller for the long interval. Vice versa, these neurons kind of increase their rates when the sequence is slower. And there is some data in primates, kind of where people have recorded activity in motor cortex while an animal executes movements at different speeds, and you do see something that looks like this. For example, these neurons tend to fire more when you have to go quicker, and they tend to fire less when the movement has to be slower. All right, and one thing you can do also easily with this type of networks is basically now, instead of generating a smooth sequence where you move forward in time, basically you can use the system essentially as an attractor network where the input to the asymmetric neuron serves as inducing transition between states, right? So here you initialize the network in the first state, and then you have a brief input to the asymmetric neurons, and that kicks the network in the next state. Then at this point, you can have retrieval at arbitrary speeds. Basically, it's just controlled by the external inputs. All right, so I think I should stop now. Right, so basically, this is just a slide of conclusions, which everything I've already said. I just want to thank people who did this work on sequences. In particular, Max Gillette, who was a PhD student starting at Duke and then moving to Chicago, who did most of the work, and Ulysses helped a lot with the dynamical mean field calculations. And this was actually also founded by this work by NIH. Thank you.